right, folks, we are going to dive into chapter 14. Okay, so we are going to go over the experimental evidence of DNA structure, and then replication, and how replication works in prokaryotes, bacteria, versus eukaryotes, us. Okay, so the central dogma of biology is sort of this DNA, RNA, protein idea, which is, in essence, an incredibly overly simplified version of what actually happens, as we'll see in chapter 15 a little bit. But we're going to be discussing the replication element of this sort of central dogma, and I like this. It's a little more specific. So your instructions are on your DNA; they're written into your RNA. Then you have all this work going on with your RNA, and then we end up with a protein. So we knew that chromosomes were composed of both DNA and protein, but what were genes themselves made of? The old hypothesis was that genes were composed of proteins. So that takes us to the Griffith experiment. So a little background with this. First, this was an experiment where different types of cells were injected into mice, and they either lived or died. So they either were injected with a deadly bacteria, or S cells, or a harmless bacteria, R cells. In the living S cells, or the mice that were injected with living S cells, that had living deadly bacteria, those mouse died after being injected with that bacteria. For the living R cells that contained harmless bacteria, those mice were healthy. Then the scientists heat killed the S cells, which means that the cells were dead, but their genetic material was not destroyed. And then they injected those into a mouse, and the mouse was healthy. Then they mixed the heat-killed S cells with living R cells, and then injected them into the mice, and found that the mouse died. And what they found inside the mouse were living S cells. So, what happened here? The living R cells were exposed to basically only some of the DNA of the living S cells, and the dead cells of the living S cells, or of the heat-killed S cells, and somehow we ended up with living S cells. So that takes us to the next experiment, or Griffith's conclusion here is that genes can be transferred, and we call this process transformation. And this is where bacteria can actually absorb foreign material, or DNA, from their environment that can actually change both their genotype and their phenotype. So the living R cells that were harmless took on something from the S cells and were transformed into S cells. So what are we thinking the S cells gave to the R cells to make them change into those deadly S cells? Well, Avery wanted to test to see that, to prove or show, that DNA is the material that both is responsible for heredity and was causing this phenomenon of transformation. So, first, what they did was they took heat-killed S cells and mixed them, and had them remove the lipids and the carbohydrates. So we just have heat-killed dead S cells with lipids and carbohydrates removed. We know that lipids and carbohydrates are not the hereditary material. So then we added proteinases to one tube, which proteinases break down proteins. So this tube is not going to have any proteins in it. Then in the center tube, we added ribonuclease, which is an enzyme that breaks down RNA. So that middle tube is not going to have any RNA. Lastly, this tube on the right, okay, we put in deoxyribonuclease, which breaks down DNA. So that means that there will be no DNA in that tube on the right. So what did they find happened? Well, they found that if you add our cells, okay, to all of the tubes, right, the tube that didn't have DNA, we don't see transformation occur. We just see our cells. And in the tubes that did have DNA, so those without RNA or without protein, transformation occurred. So this means that DNA was present in each of those tubes, and therefore DNA is the hereditary material. So just to be sure, okay, the Hershey-Chase experiment, okay, showed how a DNA versus protein from a virus enter bacteria. So these little buggers that kind of look like crazy little robots are called bacteriophages, and they are viruses that infect bacteria. So they're teeny tiny. Okay, and they mostly consist of a protein capsid coat and genetic material, DNA or RNA. Okay, and so here we have one experiment where a phage was grown in radioactive sulfur, okay, which is going to be incorporated into the phage's coat because the coat is made of protein. Okay, so that those viruses then were allowed to infect bacteria. They were blended up and separate so that the phage coat would separate from the bacteria, and then they centrifuged the bacterial pellet. And this is what they found, was that radioactive sulfur 35 was in what's called the supernatant, or the fluid, but not the bacterial pellet. Okay, so this little gray thing at the bottom of the tube is the bacterial pellet. Then they did the exact same thing with a phage grown in phosphorus 32, or radioactive phosphorus, which is incorporated into DNA. Then the viruses infected the bacteria. They separated the phage coat from the bacteria and centrifuged the pellet. And what did we find? We found that the radioactive phosphorus was in the pellet, not the supernatant, meaning what went into the bacteria from the virus was not the protein coat, but the genetic material, okay, or DNA. So if we're looking at our components of our nucleotide, okay, we're zooming in here. So this yellow dot is your phosphate group, PO4, right? P and 4 oxygens. And then we have your pentose or 5-sided sugar here, and then your nitrogenous base, okay? You are connected by phosphodiester bonds. Nucleotides are connected to one another by phosphodiester bonds, okay? And there is a 5' end and a 3' end. We'll get to what that is in a minute. For each strand of DNA, and DNA, remember, is a double helix, so there are 2 strands. Okay, these strands run in opposite directions of one another, as we'll see in a minute, or they are anti-parallel to one another. Okay, so at your 5' end, you have PO4, or a phosphate group, and at your 3' end, you have OH, or a hydroxyl group. Okay. For your nitrogenous bases, okay, they pair together, and adenine pairs with guanine, and those are your purines. And in DNA, cytosine pairs with thymine, and in RNA, cytosine pairs with uracil. Okay, so it's either ATCG or, oh, sorry, I paired them together wrong. A and G don't pair together. C and G pair together, and either adenine and thymine or adenine and uracil. So either AT or AU. My bad. So you do need to know which are the purines, which are the pyrimidines, and which pair with which. So you need to know that adenine pairs with thymine and uracil, and in which molecule they do, and that cytosine and guanine pair together as well. Okay, you can go ahead and access that, this video here. I'm not going to show it during this lecture, but you have access to the PowerPoint, so you do have access to the video there. So, as I had mentioned, okay, your nitrogenous bases pair together. Okay, so your A's and T's are going to pair together, and remember, anyone remember the bonds that we use to pair for water molecules? Okay, if you remember, those are hydrogen bonds. Okay, and those are easily breakable. Okay, and we actually have hydrogen bonds, okay, connecting our nitrogenous bases. So our A's and our T's are held together by 2 hydrogen bonds, and our G's and our C's, or guanine and cytosine, are held together by 3 hydrogen bonds. Okay, so again, your DNA is a double helix, and we have 2 different strands. All right, so we have a left strand and a right strand, and they're anti-parallel. So that first side that we looked at in the last couple of diagrams had that 5' end at the top and the 3' end with that hydroxyl group at the bottom. Now when we put our other strand with our first strand, okay, our 5' end is going to pair up with our 3' end at this end, and at this opposite end, again, we're going to have our 3' paired with the 5'. Okay, so again, anti-parallel to one another. Something that Shargaff studied, he looked at the different nucleotides in different species and found that the base composition of DNA varies between species, but the numbers of A and T bases are equal, and the number of C and G bases are equal, which makes sense. In terms of the discovery of the structure of DNA, that was discovered by Rosalind Franklin. She performed what were called X-ray diffraction and studies to identify the 3D structure of DNA, and she actually discovered that DNA was helical, and that the molecule has a diameter of 2 nanometers and makes a complete turn of the helix every 3.4 nanometers. And this was huge this was huge information, but it was not publicized as it should have been at the time, because back in the early 1930s and 40s, there were not a lot of women in science. And so she was unfortunately not credited with the discovery of DNA, even though she is basically one of the main people who discovered actually did experiments to discover its structure. And by the time that Watson and Crick, who we'll talk about in a moment, were awarded the Nobel Prize for their discovery of the structure of DNA, Rosalind Franklin had died of radiation poisoning from the studies in which she discovered the structure of DNA. So you have probably heard of Watson and Crick, probably not so much Rosalind Franklin. we're working to try and change that in science. But they eventually, using Chargaff and Franklin's work and some others, deduced this structure of DNA, and proposed that the double helix. The reason this is so important is because it allowed for the prediction that parental DNA could serve as a template to make new strands of DNA. So they predicted what we call semi-conservative replication, which is you have your parent molecule, the strands separate, and then you synthesize a corresponding daughter DNA molecule consisting of a new parent strand or of the original parent strand and a new daughter strand. So this is one way to look at DNA replication, where you have the origin of replication, and this dark blue line here, the dark blue lines here are the original double-stranded parent molecules. Okay, and then we have the daughter strands starting to form. They're going to be the light blue, and we have these arrows that are the that are going to form the replication forks. So a replication bubble forms, and then DNA is synthesized in the 5', I think it's the 5' to 3' direction. We'll get there in a second. And so it only goes in a continuous direction in one way because the strands are anti-parallel. So you end up with here, then we end up with a daughter strand, two new daughter strands pairing with those parent strands. Okay, so now we're going to look at a little more detailed look at prokaryotes. Okay, first we have our DNA strand here, all right, and we have on the top we have the 5' to 3' end, and on the bottom we have the 3' to 5' end. Okay, we have some different enzymes that we're going to be working with. Okay, so we have topoisomerase, which is going to stop additional coiling of the DNA. We have helicase, which is going to unzip the two strands. And then we have these SSBs, or these single-stranded binding proteins, that stabilize each of the single strands of DNA to keep them open. Okay. We have primase, which is going to be adding what are called RNA primers. But primase only works in the 5' to 3' direction. So that's all well and good for one of the strands, but not so convenient for the other strand. So we'll get into what that means in a moment. So now, let me walk you through this in words first, and then we're going to go back to this. So a replication fork is going to form when helicase separates the DNA strands at the origin of replication. Topoisomerase prevents the overwinding of DNA, and single-stranded binding proteins secure the single strands to prevent the helix from reforming. Primase synthesizes RNA primers, and DNA polymerase III uses this primer to synthesize the daughter DNA strand. Okay, so DNA polymerase III is going to replace RNA primers with DNA. Nope, they're not. DNA polymerase III, uses this primer to synthesize the new daughter DNA strand. DNA polymerase I, there it is, replaces the RNA primer with DNA, and then DNA ligase seals the gaps between the Okazaki fragments, joining the fragments together into a single DNA molecule. So let's look at that again. So we have the leading strand. Okay, the leading strand, all right, is going to, so we have our origin of replication here. We have our helicase, which is unzipping our double helix, okay? And we have our original parent strand here, which is 5' to 3'. And the RNA and primase adds primers in the 5' to 3' direction. Well, that is super convenient for the 5' to 3' strand. However, so you see that strand, the leading strand, is continuous, okay? There's no breaks between the production of the new strand, all right? Here we have primase adding our RNA primers, but it only, again, adds them in the 5' to 3' direction. So it has to do it in chunks, okay? And then DNA polymerase is going to synthesize the new DNA strand from that RNA primer. And then we end up with these sort of fragmented pieces because primase works in 5' to 3' direction. And when we're on the 3' to 5' strand, we call that the lagging strand because it's slower and it lags behind the other strand because it has to synthesize in chunks instead of continuously. Okay, and then we have DNA polymerase 1 coming in and replacing your RNA primers with DNA, and then DNA ligase gluing together your Okazaki fragments. And up here, your topoisomerase is preventing your unwinding of your DNA. I'm going to skip that. So this is looking at, I'm not too worried about this. Here we go. So when we are looking at, okay, this is first our strand of DNA, okay, and we know that topoisomerase is going to help stop our DNA from winding from recoiling, okay, once we get this replication fork going. And helicase is what's going to open the helix and start replication. Primase, all right, is going to be adding our RNA primers in the 5' to 3' direction. So for the leading strand, that means continuous synthesis. And for the lagging strand, that means discontinuous synthesis, where we have primase adding our RNA primers in the 5' to 3' direction. DNA polymerase 3 is going to be synthesizing these new strands of DNA after the primers. And then we're going to have DNA polymerase 1 replacing your RNA primers with DNA, and then these are going to form fragments that we refer to as Okazaki fragments that we are then going to glue together with DNA ligase. Okay, so same thing again, 5' to 3' direction. First, we're going to start with topoisomerase, which is going to keep our DNA from unwinding, and helicase is going to untwist and separate the DNA helix. Your single-stranded binding proteins are going to secure those single strands so they don't bind back to the other strand. Okay. You have primase, which is going to be adding your RNA primers in the 5' to 3' direction. So for the 5' to 3' strand, this is all well and good, and that's going to be the leading or continuous strand. And for the 3' to 5' strand, that's going to be the lagging strand, where we're going to end up using, okay, our DNA polymerase 3 to synthesize our new strand, our DNA polymerase 1 to replace our RNA primers with DNA, and then our ligase to seal together those Okazaki fragments. Okay, so again, DNA polymerase only adds to the 3' end, so we only work in the 3' to 5' end. Okay. So, okay, whoops. Here we go. So, we have our template strand, and we have primase, which adds our RNA primers in the 5' to 3' direction. Okay, DNA polymerase 3 is going to synthesize that new strand of DNA from that RNA primer. Okay, that creates an Okazaki fragment. And you're going to have multiple Okazaki fragments because, okay, when we are synthesizing in the from the parent strand that is in the 3' to 5' direction, we only synthesize again 5' to 3'. So we create these chunks that then we are going to use DNA polymerase 1 to replace the RNA primers with DNA, and then we're going to use DNA ligase to seal everything together. Okay, so in total, okay, we have your parent DNA strands, okay, DNA gyrase or topoisomerase is going to, and or topoisomerase, depending on eukaryotes, prokaryotes, is going to be securing those parental strands of DNA, okay, so that they don't unwind, or they don't rewind, sorry. And then you have helicase, which is going to be unwinding and separating your two strands of DNA. You have your single-stranded binding proteins that are going to secure your single strands of DNA so they don't rebind together. Then remember that primase, okay, adds RNA primers in the 3' to 5' direction. So that means for the leading strand, okay, if we're adding in the 5' to 3' direction, we can just continuously add those primers and synthesize that strand, and we are good to go. But on the lagging strand, okay, we are going to have primase adding our RNA primers, and then we're going to have DNA polymerase 3 synthesizing this new strand of DNA, which is in blue here, your RNA primers in yellow here, okay? And then you are going to create these fragments that we call Okazaki fragments. And within those fragments, DNA polymerase 1 is going to come around and replace your RNA primers with DNA. And then DNA ligase is going to seal and glue all of your Okazaki fragments together. Okay? So then we do some proofreading and repairing of DNA because on occasion, we might have an incorrect nucleotide pair made. For example, an A with a C or a G with a T. Okay, these types of things are increased by like cigarette smoke, X-rays. Okay, and it happens roughly every 1 in 1,000 base pairs. So there's a decent amount of mistakes made per S phase. So DNA polymerases proofread your DNA and replace those wrong nucleotides. There's also mismatch repair of DNA that corrects other types of errors. All right. So when we are repairing, we have something called nuclease, okay, which is involved in nucleotide excision repair. All right. So if we need to replace one base with another, okay, we're going to use nuclease to remove that base, DNA polymerase 2 to replace the new base, and DNA ligase to glue it back together. Okay. Over time, as we replicate, we run into a problem in that our chromosomes actually get shorter over time. And this isn't necessarily good. You can think of it like the end of a shoelace that has the plastic around the end, and that plastic over time starts to chip away, and the end of your shoelace starts to come out and fray. The ends of your chromosomes are very similar, or your telomeres are very similar. And so your telomeres are the DNA at the ends of your chromosomes. They're not genes, but they protect your genes. And there's a specific enzyme called telomerase that actually elongates your telomeres. Um, so over time your telomerase repairs sort of that plastic sheathing or your telomere around the tip of your DNA. Okay, so here is a diagram with which to practice. Okay, that last diagram that we walked through, and we will go over this in class. All right. Have a great day.